Okay to start, Michelle?
Yes, sorry.
Okay. Hello, everyone. I'm Frank Wyss. I'm the director of the Cornell Center for Materials Research. Good morning to some of you, good afternoon to some of you, and welcome to the 2021 CCMR Symposium on High Performance Soft Materials. I want to take just a few minutes to tell you a little bit about the Cornell Center for Materials Research and put the symposium in context a bit for you. The Cornell Center for Materials Research, which I'll say CCMR, is a MERSEC, Materials Research Science and Engineering Center, largely funded by the National Science Foundation. It's a big center with a lot of activity. The heart of the center is the research. We have 3 interdisciplinary research groups, and their topics are spins and magnetic material, photonic materials, and 3D atomic membranes, which means 2D atomic membranes that fold up into the 3rd dimension. At any given time, we also have a number of seed projects, and I'll tell you about an example in the area of soft matter later. We have a big educational outreach program that actually has enormous impact across the entire country. We have a wide array of shared facilities that I'll also tell you a little bit about, and we have an industrial outreach program. I want to start with the Industrial Partnerships Program, partly because it funds the symposium itself. The main mission of the program is to foster interactions between Cornell faculty and industry. The funding is entirely from the state of New York through the Empire State Development Program. And the program is designed to match Cornell expertise in materials and instrumentation with the needs of industry. The industry can be large, can be small, can be within New York State, and can be outside New York State. There are a variety of funding mechanisms to support projects that are collaborations between industry and Cornell faculty. All of these are based on matching funds that come from the grant from New York State. And the ultimate goal is always to promote economic development. If you're interested in more information about this, you can go to the CCMR website, or if you're really eager, you could start a conversation in the chat with Michelle Vanderwall, the director of the program, or John Sinnott, the industrial programs director. Some of you may know that Cornell's motto is any person, any study. It's a university founded so that anyone could study anything. Michelle and John say, by analogy, any company, any challenge. One of my jobs as the director of the center is to brag about how productive we are in our output. Of course, as a university research center, our main deliverable is publications in journals. And you can see that at a glance even, these publications are aligned largely with the interdisciplinary research groups, our primary research thrusts in the center. You can, with the click of one mouse click, you can get ISI Web of Science to do a classification on any search. And so if you search the publications that have been supported by the center since we started this grant cycle in September 2017, you see the breakdown into these different areas. I know these numbers seem huge. We are productive, but it's also partly what happens when you have 700 users of your central facilities, which I'll tell you about. The point I really want to make is that even though it's not our main research area now, we do have significant activity in soft materials. About 20 papers a year come out of center support in the area of soft materials in journals that will be familiar to you folks. One of the ways we promote and encourage research areas that are not in the center of the activity of the Cornell Center for Materials Research is this symposium. I picked a handful of the symposium themes from the past 10 years and listed them here. You can see controlling spin at the nanoscale, atomic origami, advances in photonics and quantum optics. Those are clearly aligned with our interdisciplinary research groups, but you also see other subjects, energy storage, the last time we had it. And interestingly, 10 years ago, we had a symposium on the future of biologically inspired materials. Obviously, it would be interesting to see what was predicted then and how it compares to what we'll hear about in this symposium. And then in 2015, we had one on the frontiers of polymer synthesis. I want to tell you quickly about a project that was actually the precursor for this symposium. CCMR supports 2-year seed projects in new directions. This was a project led by Nikolaos Bouklas, Larry Bonassar, Rob Shepard, and Emmanuel Gianellis, and you see their departments there. We really support interdisciplinary research. The idea was to, inspired by nature, develop tough and resilient soft materials for applications like cartilage, and ultimately to do 3D printing of those materials. This is a figure from the proposal. And I'll tell you very quickly about a couple results that have come out of that. This is work from the Bouklas and Bonassar groups. Again, mimicking nature, they've been able to develop stiff, tough, cytocompatible materials for tissue repair by arranging, and I'm honestly saying words that I don't fully appreciate here, arranging for self-aggregation of collagen networks in hydrogels. Actuators and robots, for example, that use actuators have to handle heat load. And this is an interesting recent result from the Shepard and Gianellis groups that was an outgrowth of the seed. They showed that they could make actuators that actually manage thermal dissipation by perspiring. So very interesting, obviously bio-inspired. So I just wanted to give you a feel for some of the activity in the area of soft materials in the center in the last year or two. Let me tell you about our shared facilities. CCMR operates facilities that include over 200 instruments. The instruments are for materials preparation, structural characterization, electronic and magnetic properties, electronic transport, optical spectroscopies, electronic spectroscopies, almost everything you could think of for characterizing materials. A huge strength of our facilities is the staff. We have 11 facility staff. They have an enormous amount of experience in running the instruments, in training students and users, and in guiding people to do their research. Again, it's not our current wheelhouse, but we do have significant capabilities in the area of soft matter. We have capabilities for mechanical and thermal properties of materials. We have a new MALDI machine. The FTIR and Raman microscopies are having a lot of impact. And of course, we have X-ray diffraction and electron microscopy for structural characterization. We do welcome outside users for the facilities. Okay, and the last thing I want to say is a reminder. Michelle asked me to remind you that the poster session starts at 2:45. And we would like those of you who are from industrial organizations to vote for the 3 best posters by sending email to the email address here. Michelle will give you another reminder about those later. So best wishes. The program looks fantastic. We expect to have a wonderful symposium. And I'll turn the program now over to Nikolaos and Larry.
Good morning, everyone. I'm Larry Bonassar. As Frank said, I'm a faculty member here at Cornell in Biomedical Engineering and Mechanical and Aerospace Engineering. My partner in crime for organizing the symposium today is Nikolaos Bouklas from Mechanical Engineering here at Cornell as well. I want to start out with simply a few thanks to many people. To Frank and Michelle and Vivek and John and the rest of CCMR for giving us an opportunity to arrange this symposium to, as Frank said, talk about a new and exciting area that is resonating with a lot of folks here on campus, but that's a little new to CCMR as a whole. I'd like to thank everyone attending. From what I was told by Michelle, we have almost 200 people who have registered to attend the meeting. And to the speakers in particular, this is obviously take 2 for this symposium. We were planning on doing this about a year ago, but COVID had other plans, and we were really quite pleased that we were able to essentially recreate this a year later, and that everyone involved agreed to participate even after a long delay. So we are incredibly thankful for CCMR for giving us the chance to organize this, and for all of you for participating. My goal here is simply to give you a little bit of an outline and an overview of what we're going to do today. and then really get out of the way and let all of our fantastic speakers for the day take the program and take the podium and lead us into all sorts of exciting directions. So we were thinking about this perhaps even as much as 3 or 4 years ago. As Frank said, Nikolaos and myself and Rob Shepard and Emmanuel Gianellis, colleagues here in the College of Engineering, got together and put in a seed proposal on the idea of making what we thought of as high performance soft materials. Our thinking at that time was primarily mechanical, but obviously as we've delved into this area more and more, we realized there are a lot of other applications to this. And so I've put up a couple of examples here of some recent high profile publications, both from the basic science end, thinking about engineering interactions at the molecular level to produce soft materials that are both stiff and tough, many of them biologically inspired, to larger scale applications such as this swimming robot here that involves also all sorts of interesting actuators that is composed in part of soft materials. The whole idea here is to give you a sense of the real breadth of the levels of applications that this field is seeing currently. And so we organized this symposium to be as broad and inclusive as possible with respect to the scientific area. So obviously, where we're coming from, biological organisms and tissues play a big role in inspiration and as well as direct applications. So we're gonna have some talks today that are specifically in the bio area about things like biolubricants and bioadhesion, applications for regenerative medicine, cell-based approaches for tissue repair, biomaterials for tissue repair, and then some non-biological applications, soft robotics and active materials, again, in part biomimicry and in part simply whatever applications we can think of for these new and exciting materials. Flexible electronics is something that there's been a lot of interest here at the Cornell campus on for several years. And as always, there seems to be this connection, this symmetry between a lot of the bio applications and aerospace and defense applications, in particular, lightweight materials that are deformable, actuatable, and still tough and robust. So through the course of the day, you'll hear about many of these applications. And I just wanted to give a quick overview. You should all see this online. I wanted to kind of put our spin on this. I'm not going to go through each of the titles right now because we'll introduce all the speakers in good time. One thing that I want everyone to notice is we've also tried very much to hit different sources of information. We have participants from academia, we have participants from industry, we have participants in national labs, and this is really meant to be a true interdisciplinary event where we can get ideas and share ideas from all of these different perspectives. One quick note, at the last minute, Dr. Nikia Goulburn from the NSF had a conflict, unfortunately, and unforeseen circumstances means that she can't participate. So we're going to give Dr. Flannery and Professor Zhang a little extra time, and we'll also break for lunch a little bit early. I don't think anyone will object. So with that, I'll just put out some rules for the day. Many of these have been said before, but I just wanted to emphasize them. Please keep your microphones muted during presentation. You should have been admitted to the meeting with your microscope muted. Please, with your microphone muted, excuse me. Please keep that muted. Also, as Vivek said, for bandwidth issues, probably keep video dark as well. Nikolaos and I will be the masters of ceremony here. We will look for your raised hands and questions in chat during question time. Each speaker will have some time specifically allotted for questions from the audience, and we really encourage people to ask questions. This is where the real excitement of the symposium comes in. Please attend the poster sessions, as Frank said, and we have a mechanism, particularly for the industry participants, to vote for their favorite posters, and we will give out poster awards. We have about 9 different poster sessions. These are Zoom rooms where different laboratories have organized posters. So I think we have something like around 30 posters that we have in these virtual sessions. So please come by. Everyone's invited. In particular, we're looking for feedback on those posters. Michelle will follow up with a survey via email to get your thoughts on the topic, on the organization. We realize this is the first ever entirely virtual CCMR symposium ever. So please, bear with us. And even after a year of doing all these things by Zoom, we are very much interested in your feedback. And we are very much looking forward to everyone participating and joining the symposium. So with that, I will stop sharing, and we are pretty much spot on time for our starting the symposium with the Sproull lecture. So Dave, I'm not sure, are you sharing your computer directly, or is someone sharing your slides? If you're sharing, please share your presentation. And with that, it is my great pleasure to introduce our Sproull lecture for the day. We were very fortunate to have Professor David Mooney, who's the Robert F. Pinkas Family Professor of Bioengineering and a core faculty member at the Wyss Institute for Biologically Inspired Engineering at Harvard. Dave is a good friend. Personally, one of the reasons why I am still in this field and still doing work in soft materials and bio-inspired materials and regenerative medicine is some nice mentorship that Dave gave me when I was a junior faculty member. He himself trained in chemical engineering at University of Wisconsin before getting his PhD at MIT and pursuing postdoctoral studies at Harvard Medical School. He started his faculty career at the University of Michigan before moving to Harvard about 20 years ago. I think that's right. He's a real giant in this field, which is why we were really excited for him to give this keynote address. He's published over 400 papers that have been cited over 100,000 times. He has over 50 patents. He's a member of several national academies, including the National Academy of Engineering, the Institute of Medicine, National Academy of Inventors. A true inspiration in terms of basic molecular science, materials design, multiple applications, all the way through to clinical applications and industrial manifestations of his work. So it's my pleasure to introduce Professor David Mooney. Take it away, Dave.
Well, so thank you very much for the opportunity to participate. And it was a year delayed, but I'm glad that we're able to do this. And I'm excited to participate today and get feedback from others on our work. And I'm really looking forward to the rest of the talks today in the poster session. So I'm going to take you on a little bit of a journey today. And the intent of the talk is to both expose you to a little bit of the work that we're doing in the area of soft materials. And particularly, I'll focus all on hydrogels today. And the other purpose of the way I've constructed the talk is particularly for the trainees to recognize that where we end up in research is often not where we intend to go. And that I think a really key aspect is to observe, think, and then kind of follow the data, follow the ideas. And along those lines, what we'll be doing today is my lab historically has had a lot of interest in the use of stem cells to promote regeneration. So for example, we've recently been very interested in the idea of taking stem cells from the bone marrow, which is shown here schematically on the left, and then using soft materials, cryogels, to reconstitute bony organs in the body that allow us to reconstitute the process of hematopoiesis, or formation of all the blood and immune cells. And that's what these green cells are in the bottom. So I'll tell you a little bit about how we design materials from one particular aspect for this idea of regenerative medicine using stem cells. But then we'll wander from there and, based on some observations that were made, demonstrate how this work really focused on stem cells led us to the idea of new types of medical adhesives. And so what you're looking at in this video is what we'll talk about in the later part of my presentation today, where there's a hydrogel that's been adhered onto a beating pig heart. And you can appreciate, as the surgeon attempts to pull it off, how strongly adhesive this material is, even in this bloody and mechanically dynamic environment. So to get started, I do need to mention that I have a number of interactions with different commercial entities relevant to today's talk. I will present some work that's related to research that's sponsored by Novartis and that has been licensed by Amend Surgical. The research itself that I will describe today has largely been funded by the NIDCR and the NSF, actually through the MRSEC at Harvard University that parallels the MRSEC here at Cornell. And how I'll organize the talk today is I'll first of all describe a little bit of the how we're approaching designing materials for regenerative medicine. I'll focus on one particular aspect of the design related to the mechanical properties, and then, as I mentioned, go from there to describing how this has led us to explore other types of medical devices that are inspired by what we found from the biology. Now, when we design materials to try to regulate the behavior of stem cells and promote those cells to regenerate tissues and organs, what we typically do is we go back to the normal environment in which stem cells live in the body, which is called the stem cell niche. And that's shown in the cartoon form here. Now, these stem cells receive tremendous number of different types of signals, various chemical and mechanical signals, cell-cell interactions, interactions with the extracellular matrix, which is the material that surrounds the cells within tissues and organs. So we have a really broad repertoire of signals. And as a general approach, we are very interested in using biomaterials to organize and present these various cues to the cells to mimic the normal stem cell niche to control their function. Now, I'm not going to try to talk to you today about all the different types of cues and signals that one could embed in materials to try to control behavior. I'm instead just going to focus on one particular aspect of this information, and this relates to the mechanical interaction of the cells and their environment. And a good way of motivating why this is important is this video that shows a human cell that is migrating through a collagen matrix. And collagen is a very typical material that our body uses to form the matrix surrounding cells and organizing cells into tissues. And for those of you who are looking carefully, what you can appreciate is as this cell engages with the material and migrates within it, you recognize that the interaction is not passive, but it's actually quite active mechanically. Cells in our body are constantly reaching out, grabbing, pulling, pushing on the materials surrounding themselves. And the extracellular matrix then resists these forces that the cells are exerting. So when this basic observation was made a couple of decades ago, it very quickly led to the question of whether the cells would actually respond to the resistance they felt from the extracellular matrix. So would, in particular, the stiffness of the substrate that the cells encountered change their behavior? And this led to actually a tremendous amount of research starting about the year 2000. And actually, Yuli Wang at UMass, I think, had the first really significant demonstration that indeed cells did respond to the stiffness of the materials that they encountered and adhered. He showed that the migration of cells was dramatically impacted by the stiffness of the substrate. Jun Kong, actually in the lab when we were back at the University of Michigan, demonstrated that the proliferation and cell death was regulated by the stiffness of the substrate. And Dennis Disher showed in a really elegant study a number of years ago that the differentiation of stem cells down different pathways, for example, to become a bone-forming cell or a muscle-forming cell, was dependent on the stiffness of the substrate upon which those cells were cultured. So the answer was clearly yes, the cells do care about the resistance that they feel to the forces that they exert on their surroundings. However, all of these examples are all done in two-dimensional cell culture, but clearly most of the cells in our body are not living in a two-dimensional world, but instead in a three-dimensional world. So we became very interested in whether cells in a 3D environment would similarly respond to the mechanical cues. And we were also interested in whether this concept would extend not just to the laboratory, but also back into the body, and whether we could use mechanical properties of materials as a means of regulating regeneration. Now, to explore this, the model system that I'll use in most of the data that I'll present today are hydrogels that are formed from a polysaccharide called alginate that is typically derived from seaweed. This is a block copolymer comprised of 2 different sugar units that are arranged in blocks. And for simplicity today, I'll just refer to the G blocks or the M blocks. Now, the relevance of this block structure is first that these polymers can cooperatively share divalent cations such as calcium to form a space-filling hydrogel, as shown in the schematic in the middle, but only the G blocks actually participate in this sharing, cooperative sharing of the cations, and this will be important in a minute when we talk about the structure of the networks. Now, one can readily take these materials and manipulate their cross-linking, their molecular weight, the solids content, all the typical kind of tools in soft matter to create hydrogels of varying mechanical properties that we could then use to put cells within and probe how the mechanical properties varied the cell behavior. And some examples of this are shown in this curve on the lower right. Now, the background here is actually an AFM image of one of these hydrogels. So you can appreciate the individual polymer strands, and you can appreciate that these gels are nanoporous, about actually 98% water, where you have water filling these spaces between the polymer strands. And the cells then would be encapsulated within this nanoporous gel. Now, we chose to use these polysaccharides for a variety of reasons. One of the really significant reasons is it allows us to decouple a number of really important variables when we want to understand mechanistically what's controlling the biology. And to put this in context, we're going to take a step back. And many studies in this field are done with naturally derived extracellular matrix molecules. So for example, taking laminin or collagen from tissues, purifying it, and then putting cells in gels comprised of those molecules. And the way that the mechanical properties are typically controlled with that approach is by the polymer content. So adding more and more of the protein to form a stiffer gel. Now this works quite nicely. However, as you add more of the polymer, you also change how cells engage with the material because these natural polymers are ligands for cell adhesion. So you increase the adhesivity of the system in concert with the stiffness, and you also decrease the mesh size, which may be very important in mediating diffusional transport of macromolecules like growth factors that are really important in the cell biology. So these natural materials, it's difficult to do real cause and effect kinds of experiments. Now one can, in contrast, instead use synthetic polymers to form hydrogels, and here keep the polymer content the same, utilize polymers that don't mediate cell adhesion, but then you couple small peptides mediate adhesion, so you can keep the ligand density constant as you vary the cross-linking and change the stiffness. However, in this context then, the mesh size also diminishes as the stiffness increases. So we have coupled mechanics and diffusional issues with the gels. So a really nice feature of these alginate polysaccharides relates to this mode of cross-linking that I referenced on the last slide. When they are put in solution and a small amount of cross-linker is added, you'll get divalent cations occupying a few locations within these G blocks of adjacent polymer chains, more or less aligning the structure of the network. And then as more calciums are added, you get more crosslinks formed, but you don't change the network topology. Instead, you basically just fill a few of the holes that are already lined up. So with this system, you can keep the ligand density exactly the same, the mesh size exactly the same as you change the stiffness. So it's a really nice system to decouple these variables. And just to give you a little sense of this, here's the effective diffusion coefficient of a macromolecule, BSA, as one takes a gel made out of basement membrane materials like laminin and varies the storage modulus, and you can see the diffusion coefficient is significantly altered. But when we use alginate gels to control the mechanical properties, we can keep the transport properties largely identical across a broad range of mechanics. So the last piece of this in terms of the system is we need a way for cells to actively engage with these materials. The cells from our body do not normally engage with alginate, which is derived from polysaccharides or certain bacteria. So in essence, that material acts as a blank slate. The cells in effect don't even see it. So we mediate specific interactions by conjugating small peptides to which cells have specific integrin receptors on their surface that mediate binding. And by doing this, we can take gels that otherwise would be completely uninteractive or unadhesive for cells and make them into materials upon which mammalian cells can readily attach, bind, and pull and push, which is the key feature for today. Now, the really first important data in these investigations came about from Nate Hipsch, who you see here in the lower left. When he was a doctoral student in the lab, he now runs his own laboratory at Washington University. And what he demonstrated is that one could take a cell called mesenchymal stem cells, or MSCs, and embed them in hydrogels of different stiffnesses, and this had a dramatic impact on their fate. So if we focus on the upper left, you can see that when the cells are put in a pretty soft gel, basically we got this red staining, which indicates that these cells are becoming fat cells or adipocytes. When you put the cells into this intermediate stiffness gel, we see this blue staining, which indicates that the cells are becoming bone-forming cells. And when they're in really stiff gels, they actually didn't differentiate down any pathway. And the rest of this data is just some molecular characterization of this transition and a demonstration that the density of the adhesion ligands can also play an important role in interplay with the mechanics. So this was a documentation that the mechanical properties really did matter in a 3D context and could control stem cell fate. Now, Nate went on in a subsequent paper demonstrated that he could indeed control not just the fate of the cells outside the body, but actually bone regeneration in the body by solely manipulating the stiffness of these gels. And this is the first demonstration that stem cell mediated regeneration could be regulated by these types of physical cues in the body. And he took a small number of stem cells, he put it into a cranial defect in the skull. And the idea was not to completely regenerate the bone, but to use a small enough cell number that we could see what impact the mechanics made. And you can appreciate that when he transplanted the cells in gels that were of the right modulus, he got the most significant bone regeneration, for example, is indicated by the bone volume that resulted. So both in vitro and in vivo, he found that the mechanical properties really regulated the ability of stem cells to differentiate and regenerate tissues. However, what I've been talking about to this point in time, and the field largely really acted as if the extracellular matrix that surrounds cells and tissues could be characterized solely by a modulus or stiffness value, which in essence is acting as if these things are largely elastic. However, what we know is that the tissues in our body, the extracellular matrices that surround them, are instead actually viscoelastic, as indicated by the curve here on your, basically on the left, which shows basically for a wide variety of different tissues, soft tissues, skeletal tissues, reconstituted extracellular matrices, the values of the loss modulus and the storage modulus of these materials. And what you can appreciate is pretty much all of these fall on a single line, demonstrating that pretty much all of the materials in our body have intermediate properties of fluids and solids and are viscoelastic and viscoplastic. So the time-dependent mechanical behavior is going to be very relevant potentially as well in biology, in addition to, let's say, the initial modulus. Now, in the following slides, I'll also characterize oftentimes the effect of the viscoelastic properties by the relaxation time. And when a material is viscoelastic, if one applies a constant strain and measures the stress required to maintain that deformation as a function of time, you'll see that it will fall as a function of time in a variety of these viscoelastic tissues. In contrast to purely elastic material, you won't have any dissipation of energy. But we can then characterize by the time required for half of the initial stress to be dissipated. And basically, the shorter that time is, the more viscoelastic the material will be. So we became very interested then in how do cells actually interpret the viscoelastic properties, and do they care about this time-dependent mechanical behavior of the materials surrounding them. One really simple way to regulate the rate of relaxation in the design of these hydrogels is to control the molecular weight distribution. The higher the molecular weight, the more entanglement one gets, and the more elastic the material will behave. We can also conjugate small PEG molecules onto the polymer chains that in essence act as plasticizers and will basically increase the viscoelastic behavior. The result of this is one can readily form groups of hydrogels or families of hydrogels that, for example, we can match the stiffness to be very similar, but we can have very distinct rates of stress relaxation. Here in the bottom, characterized by the half time of stress relaxation. And for this grouping, it goes from around 3,300 down to 70 seconds. And this next set of studies in which we really began to explore the impact of viscoelasticity on cell behavior were done by Ovi Chowdhury when he was a postdoc in the lab, and he now runs his own laboratory at Stanford University, and Luo Gu, who's now at Johns Hopkins as an assistant professor. So Luo and Ovi went ahead and first took cells, stem cells, and put them inside these hydrogels. And they found something pretty interesting, which is that if the gels were identical in terms of the adhesivity to the cells, the stiffness, but the stress relaxation varied, the behavior of the cells was dramatically different. The cells in the more elastic gels stayed completely rounded up, stayed separate from each other, and really didn't do anything very interesting. But as the materials exhibited more and more rapid relaxation, the cells could basically spread within the materials, they could migrate within the materials, and they could proliferate. Now, this was dependent on the ability of the cells to exert forces on the materials, because, for example, if we eliminated the ability of cells to pull and push on the materials by taking away the adhesion ligand, these effects were gone. So we could regulate a variety of fundamental aspects of the biology. We also found that the ability of these stem cells to become specialized tissue-forming cells was also regulated by the rate of relaxation in concert with the initial modulus. So for example, if we took these cells and we put them in a really soft gel, very few of the cells would become bone-forming cells, as indicated by the lack of blue staining. If we put them in a modulus that was permissive for them to differentiate to become bone-forming cells, we got more and more differentiation as the rate of relaxation became more and more rapid. So the interplay of the modulus and the stress relaxation was dramatically important in terms of controlling the fate of these cells. Now, one question that came up pretty quickly when we started doing these studies is whether this was actually relevant to anything in the body at all. So, or was this pretty much just a laboratory finding unrelated to normal biology? So Max Darnell, who's now doing a postdoctoral fellowship out at University of Washington, and Evie Lippens in the laboratory of our collaborator, Georg Duda at the Charité Hospital Berlin, mechanically characterized two environments in the body where bone regeneration normally occurs: coagulated bone marrow following a break, and then fracture hematomas from human patients that were coming back for revision surgeries. And what they found is that these tissues exhibit, are basically viscoelastic and exhibit the timescales for relaxation that were very similar to what we empirically found to be optimal for osteogenesis. So the studies that we're doing in the lab, what we're probably, in essence, is rediscovering how our body designs materials to promote osteogenesis and bone regeneration normally. Now, to further confirm this, Max took, again, a small number of stem cells, put them in gels that were identical other than the rate of stress relaxation, and planted them into cranial defects and rodents, and demonstrated that the amount of new bone formation was much greater when the gels exhibited this rapid relaxation as versus the slowly relaxing gels of the matched modulus and adhesivity. So this demonstrates nicely that the time-dependent or viscoelastic behavior of these materials is a tremendously important signal to the cells in the body and in the laboratory. We've extended this this line of investigation into a variety of other cell types at this point in time, and demonstrated that this is seems to be a fundamentally important principle to both stem cells that promote regeneration, as well as immune cells and a variety of immune responses to materials that I don't have time to go into today. Now, an interesting aspect of this, though, is that while all the data I've shown you to this point in time has been in the context of regenerating tissues and rebuilding tissue with cells, many stem cells in the body are used not for their tissue building properties, but instead for their ability to act as pharmacies and secrete a wide variety of different agents that can be important in many biological processes. And these mesenchymal stem cells that I've been describing are one of these cells that's actually the majority of clinical trials to date with these cells have actually used them for their secretome, not their direct tissue building properties. A challenge with the clinical translation of these cells, though, has been that while there's a variety of routes of administration, intravascular is the dominant approach. And what one sees with that route and virtually all the other routes is that the cells rapidly disappear after injection. And what's typically done is they're put into solution with the carrier fluid injected locally or into the vascular system. But then within about a day, the vast majority of the cells, 99% or more, are gone. So it's hard to have a biological effect if the cells of interest aren't there very long, and that probably underlines a lot of the disappointing clinical trial data to date with these cells. Now, one could simply take these cells and put them in the kinds of hydrogels I've been describing until this point in time to try to enhance their durability and control their function. And we in many groups have been pursuing that. We've also been pursuing something more recently that I think may have some additional advantages. And that is we've become very interested whether we could maintain the delivery route that people prefer today, which was intravascular, but combine it with a hydrogel coating on the cells to maintain their durability and control their function. And this work is done by Jae-Won Shin, who's now running his own lab at the University of Illinois, and Angela Mao, who's a postdoc now over at MIT. And the approach they took in collaboration with David Waits was to use microfluidics. So the idea is we have a solution of cells with our alginate solution. We create droplets where then we get gelation of the hydrogel around the individual cells. And we get a very thin layer of hydrogel surrounding each individual cell. And so here we're just looking at an image of an individual cell. The blue is the nucleus, the red is the actin, and the green is the hydrogel halo around that cell. By doing this, we can control the behavior of the cells, and importantly, we can control their persistence. So for example, if we do an intravenous infusion of these cells, the cells are going to get, the majority of them will be trapped in the lungs, which is what always happens with this mode of delivery. But the premise is that the hydrogel coating could protect the cells from mechanical environment, from being cleared from immune systems, and prolong their ability to secrete therapeutic agents into the body. And indeed, when we take these cells and we encapsulate them and we follow the fluorescent signal from the cells, you see immediately following injection, cells that are in microgels or cells that are outside of microgels, both largely lodge in the lungs. The naked cells are basically gone 24 hours later, while the cells that we encapsulate, a high fraction of them are there. And if one looks at the halftime in the optimized system we've been recently developing, we now get about a 2-order magnitude enhancement of the durability of the cells. And so we think this is going to make a dramatic impact in terms of both allowing us to decrease the dose of cells that are needed, as well as significantly increasing their utility and their therapeutic effect. So hopefully this gives you a little flavor of how we're using mechanics of soft materials to control biology in a couple of settings. However, as we were going down this path, one of the things that became clear to us is that these hydrogels we've been using, and this is not probably a surprise to anyone in this audience, are actually quite weak materials, have a fracture energy of about 10 joules per meter squared. And this really limits their applications in many settings. Now, as Nate and some of the other people early in the lab were working on the biological aspects, Shawnhee Zhao, who will be a speaker later today, was doing a postdoctoral fellowship in the lab, and he collaborated with Jun-Young Sun, who was a visiting student from South Korea. And they recognized that this idea of viscoelasticity could potentially be important, not just in biology, but also in fundamental aspects of the mechanics of materials. And in particular, what Shawnhee recognized was that the ability of materials to dissipate stresses could create a really useful toughening approach for hydrogels. And so what he and Son proposed was that if one created interpenetrating networks that combined a covalently cross-linked network that would impart some level of strength and the ability to transfer stresses across large volumes of a material, with the energy dissipation properties of these physical crosslinks, one could potentially create a large plastic zone near a crack tip that could dissipate significant quantities of energy and significantly toughen the hydrogels. So they had proposed this. They actually went on to demonstrate this idea really convincingly. And I've always liked this video from Sean-Hee's work that demonstrates, I think, qualitatively the potential impact. In this particular study, there's a 1 millimeter thick layer of hydrogel. And this is a 1-inch steel ball that was dropped from 6 feet or 2 meters onto this hydrogel. And you can appreciate this hydrogel has incredible toughness in that it doesn't fracture following the application of this really significant stress, but is actually able to maintain its structure. So these materials exhibit, really exceptional toughness, particularly for a hydrogel. Now, this was really exciting work, and I think fundamentally very important. We didn't have a particular application in mind when we did this work, but a couple years later, Jane-Yu Lee came into the laboratory, and Jane-Yu is now running his own lab at McGill, and he was very interested in medical adhesives. And as many of you know, that while we have a variety of medical adhesives, that their ability to adhere these well on wet and dynamic tissue surfaces is a really big challenge today. So the approach that Jane-Yu took to try to solve this is he looked to biology. And he found this one particular slug, shown here in this image, that will secrete a mucus that it uses to slide around basically and move around on leaves, for example. But when it's threatened by a predator, it changes the composition of its mucus, and this allows it to adhere very strongly to the underlying substrate, even in wet and mechanically dynamic conditions. And other scientists had characterized the composition of this mucus and found that it was a hybrid network comprised of both an ionically cross-linked polysaccharide network and a covalently cross-linked network of proteins. And Jane-Yu realized that this composition actually looked a lot like the composition that Sean-Hee had earlier demonstrated gave this really significant toughening behavior to hydrogels in a broad sense. So Jane-Yu proposed to take that hydrogel system and use it as the basis for a new medical adhesive. The key feature that he needed, though, was he needed a mechanism to enable that to adhere strongly and transfer stresses back and forth to the underlying tissue. And the approach he pursued to accomplish this was to use a bridging polymer. In this case, typically we're using chitosan, though we can use a variety of materials. And so the idea is we have the tissue, we have the hydrogel, and then there's going to be this green polymer here that will have physical entanglement with both the hydrogel and the tissue, have electrostatic interactions with both, and also covalent interactions that we mediate via carbohydrate chemistry. So the basic idea is we have the hydrogel, we would coat it with this solution. In essence, you can think of it like a primer. We basically place it onto the tissue of interest. The chitosan diffuses into that tissue, mediates adhesion, and then enables stress transfer back and forth between the gel and the underlying tissue. This approach, he did demonstrate, could actually work quite well. This is just a peel test of one of these gels that's been placed onto porcine skin. In this case, he actually coated the porcine skin with blood before the adhesion to determine whether or not the presence of blood would inhibit the ability to form strong adhesion. And what you can appreciate is he's forming this TA is the tough adhesion. He's getting adhesion energies in the range of 1,000 joules per meter squared. This dramatically outperforms the industry standard cyanoacrylate or superglues by several fold. And a really key feature is that cyanoacrylate, as many of you are aware, if it's exposed to aqueous fluids, it actually does not set appropriately, so the adhesion energy dramatically diminishes in the presence of blood. But the tough adhesive system its adhesivity actually is unchanged by the presence of blood, still allowing us to use it in these kinds of bloody environments. Now, to put this in context from other materials that are out there, the tough adhesive in terms of both its matrix toughness and its adhesion energy functions in this regime. The cyanoacrylates that many of you are familiar with are over here. The PEG-based materials such as Co-Seal and fibrin-based glues that are used medically today are over in this regime. The muscle-inspired adhesives are down here. So you can appreciate that these tough adhesive provide, at that time, a really unprecedented ability to mediate adhesion and to do it actually in these wet, messy environments in the body. Now, we've been exploring the use of these in a variety of contexts. And so this takes us back to the image that I first showed you. And I apologize if you don't like the sight of blood. We're basically just putting some blood on a beating pig heart to mimic a bloody surgical field. And now this adhesive is being placed on top of that heart. And basically we will allow it to adhere. And then we go ahead and look to see if we can pull it off as an indication of how strongly adherent it is. And what you can appreciate in this primitive peel test that now is being performed when we try to pull it off. And keep in mind, we only adhered the end of it to the heart. You can appreciate the adhesion is incredibly strong. The gel itself is deforming quite a bit. That's in the region it was not adhered, but we're not pulling this gel off of the tissue. So it looks like it has some pretty useful properties as a medical adhesive. Jane Yu was also able to show that it has really nice hemostatic properties, so it can stop bleeding, for example, in the context of liver lacerations. And Ben Friedman, a current postdoc in the lab, has been exploring the utility of this in a variety of different contexts. So he, for example, is very interested in how these can be used when they're attached to tendon, either as a means of mediating tendon healing following injury, or for sustained drug delivery local to the tendon. And he's been able to demonstrate that these can be placed directly over the tendon, in this case, the patellar tendon, where it will then stay adhered for long periods of time. This is just showing some MRI imaging demonstrating placement in a rotator cuff site, where you can appreciate for out to about a month, the hydrogel adhesive stays firmly in place for an extended period of time. We're interested in whether or not, in addition to sealing off wounds, if these systems could also themselves provide mechanical cues to actively promote wound closure. And so the idea here is, during when in early life, in embryonic life, if there is a wound, it oftentimes closes without scar formation because the cells will actually become highly contractile, actively close down a wound. And so what Jane Yu had asked, well, could we make our hydrogels mechanically active so they could exert forces on a wound and actively close it? So really to mimic embryonic wound healing. Now we know that these actually adhere very nicely to the skin, as you can appreciate here from this image where we have a laboratory mouse, and two of these have been placed basically on either side. And you can see that it stays nicely adherent and will stay adherent for weeks at a time. And so what Jane Yu did to transition this from being a passive to a mechanically active system that would then contract after being placed onto a wound site was to incorporate polynipam that would undergo a phase transition as it went from room temperature to body temperature that would lead to a contraction force. And so this just shows the volume, normalized volume of some of these gels as a function of time after being placed at body temperature, and depending on the composition, and particularly when he incorporated polynipam, he could get an appropriate phase transition that would lead to significant contraction. So he then pursued placing these onto wounds of animals, demonstrated that they would undergo this phase transition, they would contract, they would exert contractile forces on the underlying tissue. And if one looked at the amount of wound closure after 3 days, control animals had no wound closure. When there was the adhesive by itself, there was some minimal amount of closure, but significant wound closure when he used the active adhesive dressing that applied this mechanical load. So, we started looking at the biology of stem cells. We ended up finding materials that we think may be very useful in terms of wound healing and dressings, perhaps passive or active mechanically. So we think that this might be a really interesting application of this idea of viscoelasticity. And then I'll finish up with just one more example of this that's very recent in the laboratory. Christina Tringides, shown here in the lower left, joined the lab about 2 years ago as we were wrapping up some of this work I've been describing. And she was particularly interested in multi-electrode arrays that are used to both record and stimulate electrically active tissues like the brain and the heart, and mapping various functional regions. So this is obviously an area of very active research and clinical activity with a wide variety of different types of devices currently used for this purpose. Now, a limitation of what's out there today is that the existing multi-electrode array materials vary really considerably from tissues that they're placed on in terms of their mechanical properties. So these are typically made of metals and plastics. More recently, some of them are being made from elastomers to make them softer, but these are all orders of magnitude stiffer than the kinds of tissues that we'd like to place them in contact with, things like the heart, nervous tissue. And furthermore, these materials are purely elastic, while tissues are highly viscoelastic. And this leads to a variety of issues, including the fact that when one puts these stiff, hard materials onto soft tissues in the body, they oftentimes cause damage. So here's one example of arrays being placed over the spinal cord and some schematics of the injuries. And so you can lead to significant deformation, damage of the tissues, fibrosis that can then not just damage the tissue, but also impede the ability to have electrical interaction between the device and the material. And if we're looking at the brain, which is the particular region of interest in a lot of our work, a very high fraction of the brain topology isn't accessible with the current systems because you have these sulci, basically these involutions, and the materials that are currently used have no way of accessing unless one is to invasively place them. That leads to a lot of damage. So what Christina became interested in doing then was developing a new approach to these types of multi-electrode arrays that would be a hydrogel-based system that would be totally viscoelastic. So it would conform to the underlying tissue. It would match the mechanical properties, both the elasticity and the the modulus and the viscoelasticity. So hopefully provide a much friendlier interface that would be amenable to longer term stability, but still provide the ability to both record and electrically stimulate. And the general design of her system is shown here on the right, where of course it has to have an electrically active conductive tracks for recording. There is an insulator that separates each of the channels, and then there is a casing that basically is comprised of the viscoelastic hydrogel. So I'll just spend the last couple of minutes of my talk. I've got a couple minutes describing how we're actually designing and fabricating these devices. So first of all, in terms of the overall idea, if we have a viscoelastic, viscoplastic material, such as one based on these alginates, it now potentially allows us to actually stay in place in the brain and conform and allow us to access this space that I mentioned a moment ago isn't accessible with the more elastic materials. So this is shown schematically here, where we have a model of a lamb brain. And if we take the alginate-based arrays and we place them on the material, they will actually flow over time. They will intimately conform to the underlying anatomy. And actually, when you remove them, you can see they still have deformed. They will go back to a flat shape over time. If one takes some of the common materials, for example, Ecoflex, it does not stay in place. It actually slides around in the brain tissue over time, and it doesn't actually conform to the underlying geometry. Furthermore, what we know is that when we take these materials with appropriate viscoelastic properties, we enhance neuronal cell function, and we minimize astrocyte or inflammatory responses of cells in the brain. Now, in terms of the electronics, the approach we're taking is again to use a hydrogel based on alginate, but we're combining it with carbon materials or carbon nanomaterials, both graphene and then carbon nanotubes. And we incorporate these into the material. And the premise is if we can get percolation of the particles throughout the network, we have a path then for conductivity. The way we're pursuing doing this is instead of creating a typical nanoporous hydrogel, we instead purposely make the material microporous through a cryogelation process, so that that creates significant pores within the material, concentrates the carbon material in the walls, and enhances our ability to induce percolation at a low solids content of the carbon material. So those of you who are familiar with percolation theory, the basic idea is that the electrical conductivity, if we have electrically conductive particles, presumably will be quite low when they're present at a low volume fraction, but as they start to interconnect, the conductivity should go up significantly. And what we find is we can get actually very good conductivity in these materials once we get above about 2% added carbon material. So we get a good percolation at a low enough carbon content that we actually don't significantly change the mechanical properties of these alginate hydrogels. So we can still match the stiffness and the viscoelastic properties of the underlying neural tissue, for example, with these materials. They are very conformable. They can be independently handled. We can even tie them into a knot and bend them 360 degrees. This just shows a handful of these tracks that we've, for example, placed on explanted brain, and you can see how nicely they conform. And here's one that basically we've tied in a knot. Now, to then insulate the individual tracks from each other when they're present in an array, we use, again, another viscoelastic material, in this case, one based on PDMS. So we have a self-healing PDMS material that's previously been described. And to get it to interface with our hydrogel, we physically entangle amine-terminated PDMS into this overall PDMS network. And then we basically form a covalent interaction between the two, very similar to the way we form the covalent interaction between our adhesives and the underlying tissues. We get very good bonding between the two, as you can appreciate that even at 1000% strain, we still have good bonding and no delamination of the PDMS insulator from our underlying hydrogel. We can, with this approach, when we construct the entire device, we can actually match the mechanical properties of soft tissues very nicely. So this is just a demonstration of kind of the current materials that are out there today, and then these new materials that we're designing, where we're looking at the conductivity on one axis, the viscoelasticity on a second, and the elastic modulus on the third. And you can appreciate all the existing materials actually do not mechanically match well. For example, the heart or the brain, which are shown here. And the materials that the devices we're creating have properties that basically overlap with these types of soft tissues. Now, in terms of the ability to use these, a really nice feature of these, because they're so soft and so conformable, we can use these in a variety of different anatomies. Here, for example, you're looking at EKG recordings where we've taken the array and we've actually wrapped it around a rodent heart. So it's actually bent almost 180 degrees. It maintains good contact and gives us excellent signals. In contrast, if we took a commercially available array and tried to do this kind of recording, we would have very poor ability to record because you'd have very minimal contact between the array and the underlying tissue. We can do this in a variety of settings. So for example, in this case, this example I'm giving you, we're also looking at using it basically in the brain, where we're looking to be able to record signaling due to acoustic input. In this case, we're looking at placing it in the lateral part of the brain, which required it to be bent 180 degrees. And you can appreciate that when we basically input the noise, we get a good recording from our array indicating we are able to monitor the electrical activity. And this work is all done in collaboration with Professor Stephanie LaCour at EPFL. So I'm going to stop here. And hopefully what I've been able to give you a little flavor is how we started out with an interest in mechanobiology. We were interested in trying to understand something about how cells responded to the mechanical properties of the materials that surrounded them. This led us to then beginning to explore how we could design materials to control regeneration in the body. So for example, It really is this upper line. I didn't really get a chance to talk about the other two aspects of mechanobiology we're interested in, but along the line, because a variety of postdocs and PhD students, really paid attention and thought about, some of the data and some of the ideas, that's led us into some unexpected paths where, for example, we're designing these types of adhesives I described earlier, or here's one of these adhesives that's been put to patch actually a hole in the heart. And you can appreciate that as we cycle the heart, that patch stays well adherent and prevents leakage. The little bit of fluid you see down below is actually coming from a pore connection in the back here. And then most recently, in the work that's impressed from Christina, we're now using this idea of viscoelasticity to design new types of multi-electrode arrays that we think will be useful in a wide variety of different contexts in the body, and hopefully lead to a new approach to designing these materials in which they better match the mechanical properties of the underlying tissues. So I'm going to wrap up there, and I'd be happy, if we have a few minutes left, to take some questions.
Thanks so much, Dave. That was wonderful as usual. I really learned a lot. So as I said in the introduction, people can either raise their hands or go in the chat. So Sarah appears to have a question. Sarah, you can unmute and ask your question.
Thank you. Thank you very much. Very nice talk. Could you please elaborate a little bit on the structure of these hydrogels? I wonder what is the pore size distribution compared to the cell?
Yes. So the typical hydrogel here is going to have pores or mesh size in the nanoscale, typically like 5, 6, 7 nanometers. One can make these if one desires, and so that's much smaller than a cell, right? So the cell is on, let's say, the 5 to 10 micron size scale. If one desires, there's a variety of ways that one can make these either have larger nanopores or microporous, which is, for example, the electrical conductive tracts that I described there at the end. So they can have a wide variety of different pore sizes, and it depends on what you're desiring. Most of the biological studies I showed you, actually probably all the biological studies I showed you with cells, that was with nanoporous gels, because we wanted to really understand how the intimate contact of the cells with the material impacted their behavior.
And is there any limitation as far as the pore size distribution and the mechanical properties of the hydrogel is concerned?
So I'm not sure what you mean by limitations, but one can certainly manipulate to some extent the mesh size of the nanoporous structures. There'll be a statistical distribution, but one can manipulate that with a variety of approaches. And then in terms of the, kind of the microporosity, one can, as I said, through a variety of processing approaches, create materials that have pore sizes, ranging from, let's say, a couple microns up to hundreds of microns. Oftentimes these materials will be quite soft. So, unless one, and I think we'll probably hear more about this from some talks later, there's a variety of altering the materials by making composites. If one wants to, for example, make them much stiffer than what they normally might be with the just the hydrogel material itself.
Thank you.
Yes.
Great. I think Yadon was next.
Dave, it's a great talk. Every time I listen to your talk, it's always something new and exciting. So I have a question on the conducting material in the gel. You presented carbons, the nanoparticles or graphene. What advantage do they have over something like an inert metallic material like gold or platinum? Have you tried metals?
Yeah, so we've tried we've done a little bit of work with metals, and we can get those to function as well with this general approach. We've done most of the work with the carbon materials because we find that we can get good electrical properties at lower loadings. So we need much fewer quantity to basically get similar levels of conductivity. And so that allows us to then actually keep the bulk mechanical properties really still be dominated by the hydrogel instead of having an influence of the additive. But you certainly can do it, and actually in some other projects we're actually looking at other basically metallic materials instead.
Great. Thank you.
Yep.
And Ben had a question.
Hey, Dave. In the adhesive work, especially in the self-closing wound healing applications, I wonder if you could comment on the biodegradation timeline of that material and whether it needs to be retrieved or how it's compatible with the wound healing time course.
Yep. Yeah. So these are all interesting questions. So the material that I presented there, and actually I'm trying to think, probably all the examples of the adhesive materials that I presented today are all actually materials that will not degrade. So they're non-degradable materials. So if one is using them on the basically on the skin, then that's fine. One would simply peel them off, or they would fall off over time. For internal use, one would expect that in most applications, a biodegradable version would be preferable. And so we actually have designed these to actually exhibit varying levels of degradation, either by using polymers that themselves are biodegradable, or by having the crosslinks incorporate, hydrolytically labile functionalities. And so we can make these gels degrade anywhere from a few days up to, a few months, or basically be permanent. So one can make them have differing degradation profiles. We haven't yet combined the degrading materials with the mechanically active materials. So there's no reason why we can't, that we couldn't do that, but we haven't done that yet.
Yeah, I had a quick follow-up on that. What's the longest kind of time frame data you have on these adhesives in vivo, right? Do you know the extent to which they're remodeled in vivo, and how, whether that, you made a great point about the matching of material properties. Does that change with time in vivo for these materials?
Yeah, so the kind of, I'll focus on the non-degradable materials, because the degradable ones are, they're softening and going away with time.
Right.
With the non-degradable materials, I'm trying to think, probably the longest that we've had them implanted has been a couple of months at this point in time. That basically in sub-Q locations, in terms of functional locations, the longest I think we've gone at this point in time is about a month. In terms, and that's probably the tendon work is where we've gone the longest at this point in time. We've actually extended those a little bit beyond what I showed some of the data here. The response seems actually very favorable. We don't get significant encapsulation. They stay adherent for that time scale. They will have some encapsulation, pretty much all materials that you put in the body will get some level of encapsulation. But it looks like having a reasonable mechanical match and allows them to stay adherent for long periods of time. Does that kind of answer your question, or
Yeah.
Maybe I'm missing?
No, you
Okay.
have absolutely. So I'm also going to relay a question from the chat. Mingling Ma asked whether the mesh size of these alginate gels change upon deformation, particularly when the calcium concentration is low.
Oh yeah, that's an interesting question. Presumably will change a little bit. So you presumably will alter the mesh size, and actually that's something that can be exploited. We had done work now, actually, about 2 decades ago, where we're looking at using these as a mechanically responsive drug delivery system. And we demonstrated that cyclic loading will transiently alter the mesh size and enable one to control the rate of, for example, drug diffusion out of some of these systems. So you certainly can do that and have some control, but you're gonna, the challenge there is that it's not so universally useful, because the mesh size is a few nanometers, and so the ability to kind of transition and have the mesh size change matter will only be important for molecules that have a size scale that will be right kind of in that range. If you're talking about small molecular weight drugs, probably doesn't make a big difference. And if they're really big things, they're going to get trapped either way, because you're probably not manipulating the mesh size by that great of a difference.
Great, thanks. It looks like we are good on I don't see any more raised hands or any more questions in the chat, and I applaud Dave for being perfectly on time. We are exactly at 10 o'clock, which is the target. So Dave, thank you again. This was fantastic. I learned a ton as always, and thanks for kicking us off with a great start.
My pleasure. Thank you very much.
So with that, I will ask Dan. Oh, Dan's way ahead of me. I will introduce our next speaker, Dr. Dan Cohen. This is a somewhat of a welcoming home for Dan, or a virtual welcome home anyway. So Dan is a triple graduate of Cornell. He earned his bachelor's, master's, and PhD in mechanical and aerospace engineering here at Cornell. After leaving